Welcome back to Clinical Hematology Reviews. Today, we'll be discussing a subset of white blood cells, granulocytes. White blood cells, or leukocytes, serve as the primary cellular defenders of the immune system in peripheral blood. In healthy adults, 5 mature leukocyte types are present in characteristic concentrations evaluated via a CBC with differential, or CBC-D. Segmented neutrophils comprise 40 to 80% of circulating white cells, representing the highest concentration in adult peripheral blood. Lymphocytes follow at 25 to 35%, while monocytes range from 2 to 10%.
Eosinophils account for 0 to 5%, while basophils are the least concentrated at 0 to 1%. As a refresher, the overall reference range for total peripheral white blood cells spans 4.0 to 11.0 times 10 to the third per microliter.
After leaving the marrow, leukocytes circulate briefly in peripheral blood before migrating into tissue spaces to carry out immune defense functions. Host immunity is categorized into 2 main arms, the innate immune response and the adaptive immune response. Innate immunity is an immediate, non-specific response driven largely by phagocytes like neutrophils and monocytes. These phagocytic cells recognize conserved pathogen features called PAMPs, or pathogen-associated molecular patterns, such as bacterial lipopolysaccharide.
Beyond exogenous foreign structures, phagocytes also recognize DAMPs, or damage-associated molecular patterns, which are host molecules released from necrotic or damaged tissues. In contrast, the adaptive immune response is mediated by lymphocytes and specialized lymphoid organs. Adaptive immunity initializes more slowly, but it is highly antigen-specific and establishes long-lasting immunological memory. Together, these 2 arms ensure rapid pathogen destruction and efficient clearance of injured host tissues.
Granulopoiesis describes the progressive maturation of granulocytes within the bone marrow across 6 morphologically distinct stages. The developmental sequence progresses from myeloblast, promyelocyte, myelocyte, metamyelocyte, band, and finally, segmented neutrophil. Throughout granulopoiesis, consistent structural changes occur within both the nucleus and the cytoplasm. Overall, cell size decreases, and the nuclear to cytoplasmic ratio changes dramatically from very high to low.
As the nucleus matures, nucleoli disappear and fine, lacy chromatin condenses into coarse, heavily clumped segments. In the cytoplasm, the background color transitions from deep basophilic blue to a neutral pinkish-tan color. Primary or azerophilic granules are synthesized first during the blast and into the promyelocyte stage and contain the enzyme myeloperoxidase, or MPO. Secondary or specific granules begin production at the myelocyte stage, imparting the characteristic pinkish-tan cytoplasmic hue.
Looking closely at the individual maturation stages, the myeloblast measures 14 to 20 micrometers and features a very high nuclear to cytoplasmic ratio. It displays fine lacy chromatin, 1 to 5 nucleoli, and an agranular basophilic cytoplasm with a distinct Golgi zone or paranuclear halo. A myeloblast contains fewer than 20 primary granules and faint positivity for MPO. Immunophenotypically, myeloblasts express CD45, the stem marker CD34, and myeloid markers CD33 and CD13.
The promyelocyte measures 15 to 21 micrometers and is often larger than the blast stage. It exhibits coarse condensing chromatin, visible nucleoli, and 20 or more prominent primary azerophilic granules. Promyelocytes are strongly MPO positive and express CD45, CD33, and CD13, but lose CD34. The neutrophilic myelocyte stage measures 12 to 18 micrometers with moderate nuclear to cytoplasmic ratio and a round or slightly flattened nucleus lacking nucleoli.
The neutrophilic myelocyte is particularly noteworthy because it is the first stage to synthesize secondary specific granules, giving the cytoplasm its pinkish tan tone. Crucially, the myelocyte is also the very last developmental stage capable of cell division through mitosis. Following division, the cell becomes a metamyelocyte, measuring 10 to 18 micrometers, which is strictly post-mitotic. Its nucleus demonstrates a classic kidney bean shape, where the indentation is less than half the diameter of a hypothetical round nucleus.
The band neutrophil measures 9 to 15 micrometers with an elongated C or S-shaped nucleus. Its nuclear indentation is greater than half the nuclear diameter, showing marked chromatin condensation at both ends without thin filaments. Finally, the mature segmented neutrophil measures 9 to 15 micrometers and possesses 3 to 5 nuclear lobes connected by delicate thread-like chromatin filaments. Hyposegmented nuclei with 2 or fewer lobes indicate Pelger-Hewitt anomaly, whereas hypersegmented nuclei with 6 or more lobes signify megaloblastic anemia.
Granulocyte kinetics are divided between functional bone marrow pools and dynamic peripheral blood pools. In the bone marrow, cells reside in either the mitotic proliferating pool or the post-mitotic storage pool. The mitotic pool consists of myeloblasts, promyelocytes, and myelocytes, which are all capable of division. The post-mitotic pool contains metamyelocytes, bands, and segmented neutrophils, forming a marrow reserve roughly 3 times larger than the proliferating pool.
Upon release into peripheral blood, mature neutrophils are evenly partitioned between 2 dynamic compartments. The circulating granulocyte pool contains 50% of peripheral neutrophils flowing freely in the axial bloodstream, which is sampled during routine venipuncture. The remaining 50% form the marginating granulocyte pool, rolling along vascular endothelium in preparation for diapetesis. Neutrophils remain in peripheral blood for approximately 8 hours before migrating out into target tissues.
Specific clinical terminology defines quantitative variations in circulating granulocytes. Neutrophilia refers to an absolute neutrophil count exceeding 7.0 times 10 to the third per microliter, or 7.0 times 10 to the ninth per liter. This elevation is commonly precipitated by acute bacterial infections, systemic inflammation, metabolic intoxication, or tissue necrosis. Conversely, neutropenia is defined as an absolute neutrophil count under 1.8 times 10 to the ninth per liter, increasing patient susceptibility to opportunistic infections.
Broad changes across all myeloid lines are described using distinct clinical terms. Granulocytosis denotes an increase in all granulocytic cell lines, including neutrophils, eosinophils, and basophils. Granulocytopenia indicates an absolute decrease in total granulocytes, below 2.0 times 10 to the ninth per liter. Severe, life-threatening depletion of peripheral blood granulocytes is termed agranulocytosis, which severely compromises patient-host defense.
The primary physiological function of neutrophils is the destruction of bacterial pathogens through a coordinated multi-step sequence. First, neutrophils marginate and undergo adherence, attaching to activated vascular endothelial walls near inflammatory sites. Next, they execute directional migration, known as chemotaxis, moving along chemical gradients toward the infectious signal. Upon reaching the target, the neutrophil carries out phagocytosis, engulfing opsonized bacteria into a membrane-bound phagosome.
Once engulfed, bacterial killing occurs inside the fused phagolysosome via oxygen-dependent and enzymatic pathways. The cell undergoes a respiratory burst, generating toxic reactive oxygen species, or ROS. Simultaneously, the MPO system uses hydrogen peroxide and halide ions to generate hypochlorous acid, potently neutralizing internalized microorganisms. Together, these cellular mechanisms rapidly eliminate bacterial pathogens from host tissues.
Eosinophil maturation parallels neutrophilic development, with the eosinophilic myelocyte being the first recognizable precursor due to specific eosinophilic granules. Mature eosinophils measure 12 to 15 micrometers in diameter and classically feature a bilobed nucleus. Their cytoplasm is densely packed with uniform reddish-orange granules that stain intensely with eosin. These specialized granules contain Charcot-laden crystal proteins, major basic protein, or MBP, and eosinophil peroxidase.
Eosinophil counts exhibit marked diurnal variation, reaching their highest concentration in peripheral blood during the morning hours. Eosinophils circulate in peripheral blood for approximately 18 hours before migrating into tissue spaces. Eosinophilia is defined as an absolute eosinophil count greater than 0.4 times 10 to the third per microliter, or 0.4 times 10 to the ninth per liter. This condition is classically triggered by invasive Helminth parasite infections and allergic disorders like asthma.
Basophil development follows the standard granulocytic pathway, where the basophilic myelocyte is the earliest identifiable stage. Mature basophils measure 10 to 15 micrometers and feature a 2 to 3-lobed nucleus that is frequently obscured by coarse granules. Their cytoplasm is filled with large dark purple to black granules containing histamine, heparin, chondroitin sulfate, and inflammatory mediators. Because these granules are water-soluble, they can dissolve during routine staining, leaving clear cytoplasmic voids.
Basophils are central mediators of immediate hypersensitivity, anaphylactic reactions, asthma, and urticaria. They express high-affinity Fc receptors that bind IgE antibodies on their surface membrane. When specific allergens cross-link these membrane-bound IgE molecules, the basophil rapidly degranulates, releasing histamine and heparin. This sudden mediator release induces tissue inflammation, marked vasodilation, and increased vascular permeability during acute allergic responses.
Today, we reviewed granulocyte development, morphology, kinetics, and function, with a focus on neutrophils, eosinophils, and basophils.
That concludes our overview of granulocytes and their role in host defense. Thanks for joining us.